Okay, so now we're ready to talk about transport of oxygen and carbon dioxide in the blood. Remember, neither one of these likes to dissolve in water very well, but carbon dioxide likes it better than oxygen. So as oxygen diffuses into the blood at the alveoli, it's going to bind mostly to hemoglobin in the red blood cells. And you need to remember what you've learned about hemoglobin from the blood. Each hemoglobin unit has 4 subunits. Each subunit has one iron ion in the middle of it, and each iron can bind 1 oxygen molecule. So each hemoglobin can carry 4 oxygens. Hemoglobin has we say it has an affinity for oxygen. It will bind it reversibly. So at high oxygen pressure, hemoglobin will hold the oxygen. At low oxygen pressure, it will let it go. We describe the amount of oxygen being held by hemoglobin as a percentage. If each hemoglobin is carrying 4 oxygens, it's full. It's 100%. And we say it's saturated because it's full. If it's carrying 3 oxygens when it could carry 4, we say it's 75% saturated. So with 2 oxygens, it's 50%. With 1 oxygen, it's 25%. At rest, you usually are going to have deoxyhemoglobin with 75% saturation. In other words, you're only going to deliver 1 out of 4 oxygens to resting tissues, and that's 5 out of 20 milliliters of oxygen for every 100 milliliters of blood. So the degree of saturation is very tightly correlated with the oxygen pressure. So if you remember, oxygen pressure in the lungs is about 100 millimeters mercury. It's 100% saturated. 100 is 100. At 40 millimeters mercury, which is the resting oxygen pressure in systemic tissues, oxygen saturation is 75%. But as oxygen pressure drops with activity, your oxygen saturation drops faster. So the more active the tissues, the more oxygen you deliver. The affinity of hemoglobin for oxygen is also influenced by carbon dioxide, pH, and temperature, and a chemical called BPG. So these are the things that are going to be correlated with activity. Of course, the more active you are, the higher your CO2 level. CO2 goes up, pH goes down, temperature goes up because of activity. Red blood cells produce BPG because they don't have mitochondria. We're not going to get into the chemistry of that. Just try to remember that BPG correlates with red blood cells in active tissues. More BPG means more active tissues. And all of those things are going to make hemoglobin give up its oxygen more easily. In the meanwhile, carbon dioxide, of course, has to be moved from the systemic tissues to the lungs. So in the tissue, carbon dioxide is loaded into the blood. About 23% of that is actually chemically going to attach to hemoglobin as a molecule called carbaminohemoglobin. 70% will get converted to bicarbonate by the enzyme carbonic anhydrase, but carried in the plasma, and physically dissolved in the plasma, about 7%. This does affect the pH of the blood. Anytime you add carbon dioxide to the blood, you lower the pH. So when you lower the pH, you're going to buffer the pH change. Hemoglobin is going to buffer pH change inside the red blood cells. Plasma proteins, especially albumin, are going to buffer pH change. But even with buffering, you're going to have some lowering of the pH. The more CO2 you have carried in the blood, the lower the pH. The lower the pH, the more actively you are going to ventilate. Okay, and everything we did in the tissues we're going to reverse when we get to the lungs. So CO2 is going to diffuse out of the blood. The bicarbonate diffuses back into the red blood cells. The carbonic anhydrase converts it back to CO2, and the CO2 can diffuse back out. So that's it on blood transport in the lungs. I'm going to go ahead and finish up with regulation because it kind of goes with the blood transport. It's that relationship between carbon dioxide and pH that regulates respiration. If the pH in your brain gets too low, it just shuts the brain down. Acidosis inhibits neuronal activity. So you can actually go into a coma as a result of acidosis. So the brain protects itself very quickly by trying to maintain normal pH by ventilation. This is called a respiratory compensation for acidosis, and we'll talk about it in more detail when we get to the acid-base balance chapter. But for now, you need to remember when CO2 goes up, pH goes down, and you're going to increase ventilation to try to correct that, to get rid of the CO2. So you hyperventilate, and hyperventilating means you blow off all the excess carbon dioxide. When carbon dioxide drops, pH goes back up, keeps the pH in the normal range. And that's more important because of the activity on the brain. The other thing is that, remember, at rest you only use about a quarter, 25% of the oxygen you're carrying. So if you have extra oxygen anyway, you don't need to hyperventilate just to bring in more oxygen. Usually the problem is going to be the carbon dioxide, not the oxygen. And of course there are exceptions to that, like with carbon monoxide poisoning, but that's not what we're talking about. We're just talking about regular ventilation. So if you have excess carbon dioxide, the term for that is hypercapnia, and if your oxygen is too low, that's called hypoxia, and both of those go together. So imagine just sitting there holding your breath. You're not breathing in more oxygen, but you're using it, so you're becoming hypoxic. At the same time, you're not getting rid of carbon dioxide, so you're becoming hypercapnic. So remember that we had chemoreceptors. We have them in the aorta, we have them in the carotids, right at the base of the jaw. These chemoreceptors are the peripheral ones. We also have central chemoreceptors right in the medulla oblongata. And the peripheral chemoreceptors are very sensitive to carbon dioxide and pH, but they'll also respond if the oxygen is low. The central chemoreceptors are the ones that are just protecting the brain from acidosis, so they respond to carbon dioxide and pH. These effects are going to be not only on respiration, but also on heart rate and blood pressure. So you can imagine if you need to increase ventilation to the lungs, that you also need to increase blood flow to help carry the extra gases to and from the lungs. So anytime you stimulate those chemoreceptors, you increase ventilation, but you also increase stimulation on the heart and the systemic arteries. And all of that is sympathetic effects, along with the ventilatory effects, which are not strictly sympathetic, but they they're going to support your sympathetic response. Okay, the respiratory centers are found in the medulla and pons, and I've seen a lot of variability in what these are called and what they do, so we're just going to stick to the very simplest story. In the medulla, you have a dorsal respiratory group on each side, right and left, and you have a ventral respiratory group on each side, right and left. And the dorsal group goes to the phrenic nerves, to the diaphragm, and to the intercostal nerves, to the external intercostal muscles to let you inhale. So dorsal group for inhaling. The ventral respiratory group stimulates active respiration, so it also stimulates the internal intercostal muscle so that you can forcibly exhale. In the pons are two somewhat distinct areas that sometimes get clumped together, and they're going to regulate respiratory rate and depth. The pneumotaxic center and the apneustic center, and we're not going to worry about who does what and where in the pons. Just know that those respiratory centers are there and that they're going to regulate the rate and depth of respiration. Remember that the muscles of respiration are voluntary muscles, so if you cut the phrenic nerves, your diaphragm's not going to contract. Likewise, if you crush the spinal cord at the area where the phrenic nerves arise, you're not going to get the signal to contract, and you're going to have respiratory paralysis. So whether the damage is in the brainstem, the spinal cord, or the nerves, the effect is the same. You won't be able to ventilate yourself. So what we find is that the phrenic nerves attach to the spinal cord at about C2 to C6. And so if you have spinal cord damage between, say, C3 and C5, then you've cut off the connection between the brainstem and the phrenic nerves. And of course the intercostal nerves come off the thoracic region, so that's even lower down the spinal cord. So damage to the cervical region typically means a patient will be on a ventilator. The effect of stress on ventilation, stress, can cause hyperventilation, Bronchodilation is a sympathetic effect. Hyperventilation is a respiratory effect from the brainstem. And the specific kind of receptor that stimulates the smooth muscle in the wall of the bronchioles is beta 2. Now remember, this is vasodilation, so that's strictly speaking an inhibitory effect. Probably the most confusing thing about this section is to remember that the respiratory muscles, because they're voluntary skeletal muscles, are not sympathetically or parasympathetically controlled. So even though the brain center is connected to the same center that provides sympathetic outflow to the heart and the vessels, the skeletal muscles of respiration are controlled through somatic motor nerve fibers. That's the end of our review. I strongly encourage you to use the Interactive Physiology CD or website along with reading your book. Don't forget your homework should help as well. Best of luck on the test.